Hello, hello everybody. Thank you very much for coming to today TSVP talk. So today we have Alberto Perez, whom I'll introduce briefly. So Alberto did his undergrad and PhD in Barcelona. Then he moved to the US to work on the protein folding problem with Ken Dill. And then since 2008, he leads his own research group at the University of Florida, currently associate professor and associate director of the Quantum Theory Project. And today he will be talking about the AI in the context of structural biology. So yeah, thank you all for coming.
Hi everyone, and thank you for being here. When I asked who will come to the talk, I wasn't sure if anyone would show up, so it's really energizing to see so many people here. I'm here for 3 months. I've stayed already 6 weeks, so I have six more weeks to go. So if you like any of these, please come talk to me afterwards. What I thought I would do today is give you an overview of my field, the kind of things we do. So I'm a computational structural biologist. I work in trying to understand biomolecules, how they work, how they interact with each other. And in our field, as in many other fields, AI has made a huge revolution. So I'm going to try to tell you how we're going from protein structure prediction to molecular design. And one of the things that I came here to Okinawa to do is to try to understand better how enzymes work, how they are able to perform their work, how can we design novel enzymes that work better and do things that we desire them to do. And so the big idea about proteins and biomolecules is that there are many of them, of many different types. This is an artistic representation of what might be going on inside one of our cells. So you have multiple different kind of proteins. They are diffusing around. They're interacting amongst themselves with other molecules, membranes. There are some small molecules. There are some large molecules, molecular motors and whatnot. And so the big idea is that inside of our cell, there's a lot of activity going on. We have a lot of these biomolecules, proteins and whatnot, that in order to satisfy their functional role, they need to interact with each other. And so these molecules, they are small. So this is a cell. Proteins are about 5 to 10 nanometers. We cannot see them with our naked eye. They interact with all kinds of molecules. Some of them, like aspirin, are much, much smaller. And in order for us to understand how these molecules work, we need to know what they look like. So knowing what they look like means understanding where every atom is placed. And to do that, we have some experimental techniques such as X-ray crystallography. So we try to crystallize proteins, bombard them with X-rays, and then where they diffract, that must be the places that they collide with the nuclei. And so we can back map what the structure of a protein looks like. We can also use NMR. So trying to use nuclear magnetic resonance in order to understand what atoms might be close to each other and trace out what the structure of the protein is. And finally, we can use cryo-EM. So since the resolution revolution, there has been like a massive amount of structures. There's a lot of expertise here at OIST about cryo-EM. And so those are the 3 main techniques by which we can know how proteins look like. The challenge is that these 3 techniques, they are expensive. They might not be amenable to every protein. And so there might be other ways in which we can predict or try to understand what the structures of these proteins look like. And so for someone that as a kid liked to play with Legos, proteins are the ultimate Lego. So they are made of 20 different building blocks, amino acids. And the way that they're made is like bits on a string. So you're putting different amino acids one after another. Each amino acid, so each different color represents a different kind of amino acid. You can divide them in 4 major classes. So blue are positively charged amino acids. Reds are negatively charged amino acids. Green are polar, and white are hydrophobic. They like to shy away from water. And so under the influence of the environment and the properties of these amino acids, some of these protein sequences can undergo a transformation. They can evolve. And so I'm going to represent this string of amino acids with this string. And I'm going to highlight here 4 residues. These are 4 hydrophobic residues. They like to shy away from the water. I'm going to put in this string 4 different places in yellow. I'm going to explain that later. And one of the pieces in blue. And so what I'm going to show you is a process that we call process of protein folding. So by looking at the interactions between the different amino acids and looking at the interactions with the environment, you have every now and then some stochastic kind of interactions that ultimately lead to the folded protein. So at some point, those 4 hydrophobic residues that shy away from the water, they interact with each other, they clump together at the core of the protein, and they're protected by other residues that are polar and interacting on the outside. So this is a folded protein. You see that the 4 different yellow pieces, they are interacting together. They form what we call a beta sheet. And this blue region here, it's making an alpha helix. So this is how this protein folds. You have many different kind of proteins. They come in all shapes and sizes, and they do different things. So you have some proteins that are enzymes. They're able to break and make bonds. You have proteins that are able to recognize other molecules, maybe in disease. You have proteins that, for example, allow your muscles to contract and extend. And you have membrane proteins that allow things to go in and out of the cell, either molecules or signals coming in out of the cell. So one of the things that we want is to understand what the structures are like so that we can understand what are the functions of these molecules. And so I've shown you the process of protein folding. There are 2 kind of competing interests. On one side, you have the chain entropy. So every time I put like something inside my pocket, it comes out like a tangled mess. So that's the entropy. And on the other side, you have like the interactions that these amino acids can make with each other that favor folding. And so you have an entropic term, an enthalpic term, and there's a balance between the 2. For some protein sequences, what you find is that at low temperature, you can find this native state, this folded state. And this is a funnel-like representation where at high temperature, you find unfolded states. At low temperature, you find the folded state. Now, how many protein conformations, how many conformations can a single sequence adopt? Many. Many, many, many. Many, 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 many. In fact, more number of possible configurations than the number of hydrogens in the known universe. That's how many possible conformations every single sequence can do. So if a protein had to go through every single possibility before finding a native state, it would take longer than the age of the universe for a protein to fold. Obviously, we cannot wait that long. And so our proteins fold very fast. The fastest proteins can fold in milliseconds and even in microseconds. And so that's how fast they fold. And so for the longest time, this led to some questions in our field. It's what's called the protein folding problem. It's a 60-year-old kind of problem, and it has 3 parts. The first one is, how do proteins fold so fast? What are the structures that they adopt? And third, can we use computers to predict those structures? And so these last 2 questions have been mostly solved recently, and I will tell you how. This third question, how do proteins fold so fast? That's still something that we are discovering. So I've told you this is a 60-year-old kind of question. So for the longest time, if you look at papers, historical papers, you will see, oh, we solved the protein folding problem. And then someone else published another paper, oh, we solved the protein folding problem. It turns out like, yeah, maybe they did something with 1 or 2 proteins, and then you try to apply the methodology to a new protein, and it didn't work. And so one of the biggest contributions to our field is the emergence of blind prediction competitions. So these are events in which someone is going to say, well, we're going to start a competition. Everyone in the world can participate. What we're going to do is we're going to contact some experimentalists that are working on crystallizing or solving with KRM some of the proteins. We're going to ask them for the sequence. And then we're going to provide this sequence to everyone who wants to participate. So this usually happens every other summer. So during summer, we have 2 months. Every day, we're given about 2 sequences. The competition is going on right now, actually. And for every sequence, you have about 3 weeks to predict up to 5 structures. So you submit your 5 structures, and at the end of the competition, someone who has not participated will be assessing all of the predictions. This is what we call a double-blind experiment. Blind to the predictors because we don't know what sequences we're going to be given. So our methods cannot be geared up for a particular sequence. And then it's blind to the assessors because they're given all the predictions, but they don't know who predicted what. And so at the end, what you get is what is the state of the art of the field? What are the best methods to work on the protein structure prediction problem? How do you think they did on the first chance? Good, bad? Bad, right? Yeah, so the first experience was a cathartic experience. So if I were to go back in time to 1995, and I were to say, well, this protein has all the atoms on top of each other, I would have one cusp. That doesn't have any physical kind of sense, but according to the metrics that they use, that would have been the best protein. That's how bad we were at predicting proteins. All right? Did we get better over the years? Well, this is a plot that is going to try to explain that. The answer is no, we didn't get much better. So each line here is a different competition. So every 2 years, we have a new line. On the y-axis, you have the ability to have accurate predictions. So good means the atoms are in the right place. Bad means it's random noise. And so on the x-axis, I have whether the protein sequence that we were given is easy or hard. How do you tell if a protein is, or a sequence is easy or hard? Well, I'll tell you a little anecdote. So I came to Okinawa. I'm originally from Spain, and I was walking around, and I heard something that sounded like a duck. I thought, I went around, I looked for it, and it looked like a duck, but not like any duck that I knew about. And so I said, well, it sounds like a duck, it looks like a duck, it must be an Okinawan duck, right? So they do the same for protein sequences. If a protein sequence looks like another sequence for which I know the structure, I'm going to say this sequence most likely falls to that structure. So that's how we define an easy sequence. And for easy sequences, we get good structures. For proteins that don't look like other proteins, their sequence, we have very bad ability to predict their structures. So, all right, so over 20 years, almost no progress. What did change is how much data we accumulated. So back in the 1990s, we knew about 50,000 sequences. Then came all the genomic and metagenomic projects. We now know about over 200 million protein sequences. We used to know about 4,000 structures deposited in the protein data bank. We now know about 300,000. So this changed from a problem of like, oh, can we use physics or can we use statistical mechanics to predict the structures of proteins to an AI kind of challenge. And so in my field, computer programs and science have gone hand in hand. So in the 1990s, IBM developed Deep Blue. It beat the reigning champion, Kasparov, at the game of chess. So basically, the idea is that by brute force, a computer can calculate more moves than a human can. At any point in time, you can look at the chessboard and you can decide who's winning. And so with that, Deep Blue beat Kasparov. So IBM said, oh, now we're going to solve protein folding. And they didn't. So a little bit later, a company named DeepMind, is a subsidiary from Google. They decided to play the game of Go. Now, very, very typical game in Japan. And the idea is that here you're putting stones, either white or black, and you're trying to capture the pieces of the opposite side. The problem is that there are many more possibilities than in the game of chess. So now you cannot just by brute force predict who's going to win. And there's no way to know who's winning by looking at the board. You really have to play out the game. And so if we were just waiting for a computer to be fast enough, we would still not be able to beat the reigning champions. And yet, AlphaGo was able to defeat the world champions. And so, and one of the particular interesting things, so this was 5 matches. I think it won 4-1. And in one of the matches, the commentators, there are commentators for these games, said, oh, the machine has gone crazy. It has made a move that makes no sense. 100 or something turns later, it turned out that that move was critical to win the game. And so it had hallucinated a move that no human would ever make, and yet it was critical to win the game. And this is something that will come back later. Because when AlphaGo won, they said, well, our next challenge are proteins. And so I've shown you that there was no progress in 20 years. AlphaGo came into so AlphaGo changed to AlphaFold. And in 2 years, we went from here to all the way here. At this point, they were just newcomers to the game. They were just doing everything that everyone else was doing. So they had more resources, they had more people working on it. So in a few months, they were able to beat the whole field. 2 years later, they said, well, we can now develop new transformers that pay attention to the sequence and structure similarities. And now, this is the state of the art. And this technology was actually the one that recently won the Nobel Prize in 2024 for structure prediction. And so, of course, I was at this conference where they announced the winners. I came back and my lab was like, oh, why did I ever join your lab? Like, this is gonna be a disaster. I'm never gonna get a job afterwards. This is horrible. And of course, me being the optimist that I am, I said, don't worry, guys. A structure is just the beginning. So we have AlphaFold. It's a great new tool. Let's use it to be able to do new science that we couldn't do before. And so the big thing is, like, if I go back to the duck that we saw earlier, you see the duck with the wings tucked in, and you never know that a duck can expand its wing and then flap them and start flying, right? So knowing something about the structure or one state doesn't tell you anything about the function. If I show you this protein called ATP synthase, so what? Yeah, it looks colorful, it looks nice. What does it do? Well, this protein is actually a motor that it captures. It's a hydrogen pump, so it captures hydrogens, and then it's able to make ATP, the molecule that allows you to carry energy in to do many of the reactions in the body, in the cell. And so it is critical to know the structure, yes, but that's the start to be able to understand how these molecules work. And one of the key aspects of how these molecules work is the concept of molecular recognition. So if you think about your daily life, so this morning I was on my biking here, then, right now I'm holding this, I might be holding a pen, interacting with a keyboard. So in order to do different things, we're interacting with different objects. In the same way, proteins, in order to carry out their work, they need to interact with other molecules. Some proteins will interact with nucleic acids to maybe express the information in your genes. Some will interact with other proteins or peptides or even small molecules like aspirin. And so understanding molecular recognition is key to many fields. In biomedicine, to design novel molecules that can stop disease. In biotechnology, to design custom-made sensors or possible enzymes that can catalyze reactions that we care about. Biomaterials to do, for example, self-healing. You put two peptides together, they form a hydrogel that can capture cells to close a wound, or to design novel functionality that we have never seen because evolution never thought of it. And so if I'm thinking about molecular recognition, it goes between two extremes. On one end, you have something that is called lock and key kind of interactions. So this is where you have a molecule, it might have like a cavity or something where a small molecule can bind. And on the other hand, you have what's called dynamic recognition. So proteins that might be intrinsically disordered, they can adopt many shapes. And once they find the right binding partner, they adopt very well-defined shapes. So if I had to put a cartoon representation, this is kind of what I thought about. And on the other side, you have your hands, which can adopt many different conformations depending on what they're interacting with, right? So one of the things is knowing a structure is not enough. Now we need to understand how this works. So for a lot of the things that I do in my lab, like we work with physics-based models. So in physics, like you try to put a physics model, you use the equations of motion, Newton's equations of motion, you propagate them through time, and you see the wiggling and jiggling of atoms. And if you look at this for long enough, you might be able to understand the behavior of biomolecules. The only problem is that these simulations are computationally expensive, very, very expensive. So Right now, about 20 to 30% of supercomputer time is devoted to these kind of simulations, and it produces terabytes of data, and yet understanding what these molecules do or reaching timescales that are relevant is hard. And so how do we predict binding? So I'm going to tell you about the two different regimes. The lock and key regime. So how do we predict binding for a small molecule like aspirin? And how do we predict if we have many different compounds, which of them might be the one that binds best? And so with physics, it's very hard to predict things in absolute terms, which one is the best. But if I give you something like this horse, so if I give you something like this horse, it looks fast. This horse also looks fast. But if I were to say which one of them is faster, the best way is to do a horse race, right? And so we do exactly the same with our molecules. So when we have this lock and key interaction, we compete different compounds in order to understand which one of them might have the best binding affinity, bind the strongest. And so this is something that we use routinely in pharma. So in drug discovery, this is the early stages of drug discovery. It's widely used. You can process thousands of compounds, no problem. On the other hand, when you have flexible molecules, now that becomes a problem, because not only you have to care about what molecules are interacting with which ones, you also have to care about what are the different shapes that they might adopt and what is the right one. And so it's a much more complex problem. So we've worked in this area, we've developed some methods based on physics, and basically the idea is that we have a protein target, we have two possible compounds, they are disordered when they're on their own, and they might adopt like a helix-like structure when they bind. So we can make these comparisons. It takes us about 30 GPUs running for 5 days nonstop in order to make one of these predictions. Now imagine that I have 100,000 of these compounds to test. There's no way I can do it with a computer. So as soon as AlphaFold came out, what did we do? We said, well, you know, they designed AlphaFold to predict the structures of proteins. But a peptide binding to a protein, that's a similar kind of problem. It might work. And so people started using AlphaFold for all kinds of things that it wasn't designed to do. It turns out that it did pretty well. It did really well at predicting protein-protein, protein-peptide, and other kind of interactions. It was also really good, not only at predicting structures, but people started doing things like flow matching and whatnot to predict ensembles, predict different conformations away from the static structure. They also used it to design novel proteins. And so from AlphaFold that was designed to do only one thing, a lot of different functionality emerged. And that has opened the field to many, many different kind of things. So still, we were like, okay, but we don't care only about knowing where something binds. We care about knowing which of these compounds might be the best. And so we said, if I give you all of these little compounds that go from strong binders to weak binders, will AlphaFold know how they bind and which ones are the strongest? So it turns out that AlphaFold did very well at predicting that all of these bind, but it could not tell us anything about which of them was the best.
Can I ask a question? So these molecules live in some environment, aqueous or something else. Where is that information in here?
So in the AI model, that's implicit because it's been trained on either NMR structures, which already capture some environment, crystallographic environment, cryo-EM environment. So they capture whatever environment they were in, right? In a physics model, we would explicitly put the environment, either a membrane environment, either an aqueous environment with ions, a crowded environment, so on and so forth.
Thank you.
So AI is very confident about these different peptides should bind, but we have no idea which one of them is best. And so if I were to ask you, do you like Japanese snacks? If I ask you about this snack, do you like it? Pretty confident, yeah. Do you like this one? Yeah. How about all of these? I'm pretty confident you like all of these. But being 100% confident on each one of them doesn't tell me anything about which one you would choose, right? So confidence is not the same as preference. So we said, well, if AlphaFold is really confident about all these predictions, could we ask it something about which one to choose? And so we started thinking, so my student Liwei, he started thinking about like, well, maybe in this function that AlphaFold is encoding, it has learned something that looks like a scoring function. And could we, instead of predicting one peptide at a time, could we feed it 2 peptides at a time? And now it has to make a decision. Which one are you more confident with? This one being in the active site or the other one? And by doing that, we could predict which peptides bind and which ones are the strongest. And the big game for us is that when we use physics, we take 30 GPUs running for 5 days. When we do this AI approach, it takes us 1 GPU running for 5 minutes. And so now we can scale to hundreds of thousands of compounds. So what that means is we can go get whole protein libraries, screen those protein libraries, cut them all into all their little pieces, and identify which ones bind, which ones do not bind, and which ones are the best. So we're working with some immune response proteins. We're now understanding many proteins that are able to bind these immune response proteins, from viral proteins, from bacterial proteins, and even host regulatory proteins. And so this is allowing us to learn many new things. Now, one of the things that we are seeing is like, well, AI still has its own information. It's fast, it's really good, but it might have some biases. So what our new paradigm is that we're combining the speed of AI with the interpretability that physics-based models are giving us. And with that, we're now able to make more predictive molecular modeling. So basically, we have a front end in which we use AI at the sequence level, then at the structural level, and then we go to increasing degrees of complexity of physics-based models. When we are done with all our filters, we go down and we send our predictions to our experimental collaborators. And with that, we have been able to identify novel proteins. And we have also been able to distinguish some proteins that were previously characterized and were not biologically relevant. All right. So far, I've told you about protein structure prediction, the problem of molecular recognition. And then the next stage that I want to tell you, the last part of this talk, is about going from a structure prediction to design, right? So the idea of design is the inverse problem as protein folding. So in protein folding or protein structure prediction, we have a sequence, and we want to know what is the 3D structure that it adopts. And that has been mostly solved by AI. In protein design, what we want to know is if I have a 3D structure of a protein, what is the sequence that is best suited to adopt that structure? And so the structure could be a structure that it's already known, or it could be a novel structure, a structure that is hallucinated, for example, by AlphaFold. And so the main idea here is that during evolution, things evolve until a certain point. So you have some parameters. For example, most of our proteins have to be at least stable at 37 Celsius. When you have a high fever, you are putting some cold ice on your forehead or whatnot to lower the temperature so your proteins don't unfold or whatnot. And so with designed proteins, you can now go to proteins that are stable, maybe 80, 90 Celsius, whatnot. And so one of the biggest players in the field is David Baker. For the longest time, designing proteins was a task that very few groups in the world could do. David Baker was the maximum exponent of what we could do. And before AI, they were already doing very impressive things. So this is one of the designs that they did. It's basically a protein that is able to fold into a 3D structure. It's able to assemble itself with other proteins of its type and make this cage-like kind of compound. So you have 40,000 atoms here that when you solve the experimental structure, those 40,000 atoms are on top of the design. That's amazing. So the challenge is that only 1 or 2 groups were really good at doing this. The success rates were not high, and it required a lot of manual intervention. So it was very artisanal in some way. And so the idea is, could AI help in this front as well? And so from 2015, where Baker Lab was the maximum exponent, to today, what we're seeing is that we have now democratized design. So basically, any lab can now, within 1 or 2 days, say, hey, I want to design a protein that binds this particular target. And within 2 days, you have 10,000 designs. Now, are all those designs good? No, you still have to find out what are the best. The success rates are 10 to 20%, and programs are getting better and better. But like having like this AI machinery has helped a lot. So this is the Nobel Prize. 50% went to David Baker for design, and 50% to Demis Hassabis and John Jumper for structure prediction. So how does the design work? So it works pretty much the same way as in the image generation field. So we use what's called diffusion models. So the idea is that if I have a picture of a dog, it's made out of pixels, right? I can randomly change a few of the pixels in this picture, and I will get something that still looks like a dog, but not quite. I can learn the process of going from this picture to this picture, but also from this picture back to this picture, right? And I can do that again. And I can do that again and again and again until I get to a random set of pixels. Now, if I have this set of pixels, how do I change them to get a new image? I have no idea. But if I have a random set of pixels, I know how to generate another random set of pixels. And if I've learned the process of going from a random set of pixels back to an image, I can say, well, this is my new set of random pixels. And if I go back, ooh, now I get a cat. So of course, to do that, you have to train on many, many, many images. Yeah? So I can do exactly the same with proteins. I have about 300,000 proteins. I know the position of all the atoms in the protein. I can add a little bit noise to the positions of those atoms, and I can learn that step. I can also learn the step of going back. And I can go all the way until I get a random distribution of atoms in space. And from that random distribution, I can generate a new random distribution, go back and generate a new protein. So that's how design works nowadays. Once I have the fold, I calculate what is the sequence that is best suited for this protein design. And so again, like we've been using the latest AI technology combined with our physics-based pipeline. So basically we start, we use diffusion models in order to predict new proteins that will bind a particular target of interest. Then we do all of our AI pipeline to filter out which we think are the best designs. And then finally, we use our physics-based models to say, like, do these two things that have been completely orthogonally designed, do they agree or do they differ? When they agree, we send those for experimental validation. And this has been successful in many cases. I started the talk by giving you an example about blind structure prediction. There have been new challenges for designing proteins. So this was one that we participated a little while ago. It's called the BioMel Challenge, Pitch to Binders. So the idea is they gave us this target. So this is a cancer cell. It has a tumor antigen. And usually a T cell will have to recognize that antigen, bind to it, destroy the cancer cell, right? So they said, could you design a mini protein that would bind this antigen? And if you can, then what we're gonna do is we're gonna take that mini protein, we're gonna put it in a T cell. So we're gonna have a functionalized T cell. This is called a CAR T cell. And so once we have that, what we're gonna ask is, can those designs be successful at killing these cancer cells? So basically that's what they tasked us to do. About 12,000 sequences from around the world were submitted. Each team could submit 500 sequences. So once they gather all the sequences, then they did all these kind of functional assays. I have no idea about these functional assays, exactly what they are. The basic idea is they tested whether the CAR T cells were able to proliferate in the presence of cancer cells. If these CAR T cells were able to send signaling molecules, that would activate the killing of the cancer cells. And finally, what was the ability of these T cells to kill cancer cells without killing regular cells? And it turns out that our submission was the top submission in the whole competition. So it was really nice. They 3D printed awards for the top teams, and they sent it to us. So this was the mini protein that we designed, and this is how it binds its cancer antigen. And so, like, this was a nice blind competition. One of the biggest surprise is that the best binder was not the one that won. The best binders actually create exhaustion, and so it doesn't allow cancer cells to be killed. And so this was one of the lessons that we learned along the way. So by now I have told you 3 things. I've told you about protein structure prediction, about how that's not enough. We need to understand molecular recognition, how these proteins interact with different molecules. And the hope is that we can use that to understand how proteins function. So basically, this challenge of protein structure prediction is mostly done. A lot of molecular recognition is now what's going on in our field. Like, there are many things that we cannot still understand using AI models. For example, things like how do cryptic pockets emerge, or how does allostery happen? Like, those are too complex for most of the AI models that we have right now. And the future, where everyone is trying to go, is how do we look into protein function? How do we go from a sequence to function? And so with that, I just wanted to finish with a picture of my group. Really happy to be here. I'll take any questions. Thank you.
Yeah, sure. Yeah, please, if you have any questions, just please wait for the microphone for the people on Zoom.
Yeah, thank you very much. It was so nice to see such a good presentation. So I have 2 questions. The first question is about the approach when you combine AI and physics-based approach. So what is the lack of just using AI and the sequence? What's the lack there? What we take from the physics, is it just the more parameters? Because like physics is kind of embedded in the sequence, I would say, as well.
All right, so the main thing for us, so whenever you use an AI model, one of the biggest questions that reviewers can ask you is, oh, what about memorization? Like, did you get the right answer because it's making a prediction, or is it because it has already seen this, and so it's giving you the right answer for the wrong reason? If it's giving you the wrong answer for the wrong reason, then it's going to make predictions that are false positives. When you use our physics-based model, because it hasn't been trained on data, it just uses, like, how do bonds stretch, how do angles change, and so on and so forth. If the answer with our physics model agrees with the AI model, then that increases our confidence. We could still be wrong, but we're much more confident because they have completely separate origins.
Okay, and I have the second question, also short one, about the diffusion and the protein design. So are there any like common cases when it doesn't work? Or just by chance?
Yeah, so all right. So if I were to say, okay, I'm going to use RF diffusion to predict binders to this molecule, and then I'm going to design the best sequences that are able to adopt this 3D structure, the success rate is expected to be 10 to 20%. So that means one every 5 will fail. Of course, you can only test experimentally. Like, my collaborators will try at most 5, right? So if I choose 5 that are incorrect, they're never going to collaborate with me again. So we need to make sure, as best as we can, that what we're giving them is enriched from having 1 every 5 correct to 3 or 4 every 5 correct. And so that's why we do this complicated machinery. DeepMind has done a new AI that is proprietary, so this is not available to the, I think it's called AlphaFold or something like that. So this one is not available, but this one is much, much, much better at making, so it goes from 10 to 20% to a much higher percentage of success.
Yeah, my question was more about like we know that let's say basic AlphaFold doesn't work well for strictly disordered proteins. Let's say maybe is it this like the case for diffusion models as well?
So for disordered proteins, for predicting and designing disordered proteins, I don't think it's going to do very good. Like I think like there are some AI tools that are starting to predict how to bind disordered regions, but not necessarily to make disordered regions. I mean, like there are other tools that can give you disordered regions, but usually I think the state of the art right now is I have something that is disordered, I will use NMR, and I will do a population of states, reweight them so that they satisfy the NMR data. That's, I think, the best that you can do.
Thank you.
Thank you, Alberto. That was really nice to see. And so this is probably a very simple question, but still, you had in the beginning like a landscape of energies, and you talked about the native state when the lowest energy, right? And then you also had one question that was still you're working on how they can solve this hard problem so fast in reality. I guess, I mean, do they always find the lowest one, or is it no? Or I mean,
so
yeah,
first,
okay.
Not all protein sequences will have a folded state. Some of these proteins will have like a more complicated landscape where like you have like maybe a misfolded state that can aggregate. And so they find this aggregated state before they find the native state. So you have like, what are they called? Chaperones that will help fold some proteins that are misbehaving. So some of them need help. Yeah.
But eventually they will find the absolute smallest one, or?
Well, and when they don't, we get all kind of diseases, right? Like Alzheimer's, Parkinson's, and whatnot.
Thanks.
I'll be here 6 more weeks, so I'll be 2 floors above if you have questions.
Okay. Can I add one more then?
Sure.
Yeah, just also.
No, you're fine.
Yeah, okay. So I remember when they got the Nobel Prize about, because it's a statistical, method in essentially that.
I would not call it a statistic.
Okay, it's about, I remember the statement was that now we understand how proteins are folding. I mean, it's a good predictor, but it's not the explanation, right?
It's not the explanation.
It's a black box still.
So, all right. Let me backtrack a little bit. So when I said the protein folding problem is these 3 questions. So for me, the protein folding problem is not solved. The protein structure prediction problem is mostly solved.
Right.
And so depending on who you ask in the field, they will not distinguish those 2 things. For me, protein folding is understanding what are the myriad pathways that will take you from unfolded state to the folded state. Each one of those pathways will have different kinetics and different timescales, but on average, altogether, they make life possible. So it's robust enough that on average, a protein will be folded every X number of units.
Because of that understanding how that is actually the path to taking will make us maybe think about drugs that prevent misshaping, right?
Sure.
Yeah. So I think it's good to have the difference between understanding and making predictions.
Yeah.
Thanks.
So, Mike. Thank you very much for the lecture. That's like but you come from the simple explanation to the complex ideas, and that's very nice. I have a question about like you want to know how to come from the structure to sequence, and maybe why do we need it right now? Maybe we need it first to understand how the structure codes the function, and then understand how to come from the structure to sequence.
Right. It would be very so. Like, let's go back to the DAG, right? Like, say AlphaFold for the DAG, it would predict the DAG with its wings stacked. That doesn't tell me anything about what is it that the DAG needs in order to fly, right? I need to understand this other state, which is the wings open and down and up. So those are the 3 states I need to understand for DAG, right? If I understand those 3 states, and what are the kinetics between them, then I can understand how it functions, how it flies.
Yes, so understanding the like the path from the structure to sequence is the key for understanding the structure to function.
So one more time. So going from structure to sequence is key to
understanding structure, how we can define structure, function of the protein.
Not necessarily. I mean, like I think like they are independent tools, but all together we will get more knowledge to try to understand function. So what is the degenerate code of sequences that are able to fold to a particular structure, and what are the states that they are compatible with, and like how does that relate to function? That will be interesting, but I think it all plays together, but not necessarily we need these to learn the other thing.
Yeah. Okay. Thank you very much.
Yeah.
Any further questions? Well, if not, yeah, thank you very much again for the talk. And thank you for coming.